So I'm pretending today is National Pajama Day, but to let the rest of the class know, there actually is a National Pajama Day. It is on a Monday, April 16th. It's the day after Tax Day. So I expect to see you all in your pajamas that Monday. So we'll finish up this chapter and we'll get a little bit into chemistry. And the chemistry concepts are difficult, but all I ask you to do is do a lot of memorization and rewriting of some of the chemistry so that ultimately over time you'll be introduced to it more and more, and it will actually make more sense as time goes on. We were finishing up with the large intestine. We were talking about its motility. It does have a segmentation pattern because its goal is to absorb water as well as various ions that are remaining in the water, and some vitamins will be absorbed in the in our large intestine or colon. So we call those slow segmentations of the large intestine hostile contractions, and the peristaltic wave that occurs in the large intestine is actually called mass movements, and we only have about 2 or 3 a day, and typically after a meal. So there is a reflexive peristalsis that actually occurs after someone eats. And this is something to actually utilize if you have a child or a family member that has a little bit of constipation. After a meal, to encourage them to actually go to the restroom actually can kind of help alleviate some of that. Kids would rather play instead of going to the bathroom and are sometimes prone towards getting rather constipated at times. And so if you ask them to try and go to the bathroom after dinner, for example, you may actually kind of help out with preventing that process for them. Defecation is actually a reflexive pattern, and it is because baroreceptors in the rectum will actually be stretched. So we have stretching of the rectal wall because of the arrival of more and more fecal material, and the amount that is required to actually initiate the defecation reflex is kind of rather individual. Some people don't require much and other people actually require a lot more. But during the defecation reflex, you will actually have relaxation of the internal sphincter, which is a smooth muscle, and you'll have contraction of the muscularis externa of the large intestine in the area of the sigmoid colon as well as the rectum, and that will cause that peristaltic wave and cause the expellation of fecal material only if, once you're potty trained, only if you relax the external sphincter. And so the external sphincter is a voluntary muscle, and defecation will not actually occur in most people unless they actually voluntarily relax that. And most parents are really happy when their kids learn that they actually have control over that sphincter, and potty training is considered a successful event when they no longer have to wear their diapers out during play or at bedtime. We're going to touch a little bit on the physiology of digestion and absorption. Remember, digestion has a mechanical phase, breaking things into smaller bits and pieces so that we can then have enzymatic digestion to finish breaking things down into their monomers so that the cells can actually absorb that material from the luminal side. And then once the material is absorbed into the cell, then it has to be put into the soft tissue for the capillaries and the lymphatic capillaries to actually, called lacteals, to actually carry the stuff along to the liver for processing. So when I say enzymatic hydrolysis, I'm actually talking about chemical digestion here. And we want to break those large molecules into those building blocks or monomers, and CBH stands for carbohydrate. So that's something that you might add to your abbreviation that you might need sometimes when you take notes. And remember, the monomers for carbohydrates are monosaccharides, simple sugars. And when we think of the most common monosaccharide we think of is glucose, but we also have fructose and galactose. Those are simple sugars that we most commonly think of. When do we actually have typical enzymatic hydrolysis occurring? And hydrolysis, let me ask you, what does the word itself, when you hear the word hydrolysis or hydrolysis. What does that mean to you if you can break it down? Water is going to be actually utilized. So we're going to take a big molecule, we're going to cut it up in bits and pieces. The water molecule is going to come apart and be attached to the ends that we actually just cut. So you can think of the ends as being kind of a raw or open wound end, so we will seal off that end with water. So hydrolysis is a very good term to use when we actually make smaller molecules, because the water actually kind of, so to speak, seals the cut ends of the little fragments that we're actually then creating. So let's see where we actually start with carbohydrate enzymatic hydrolysis. And that starts in our oral cavity. Remember, we have an enzyme that's in saliva called amylase, and so that begins that. Things go into the stomach, but classic enzymatic hydrolysis of carbohydrates is not actually in the stomach. So various enzymes that will actually apply to the carbohydrate besides the oral cavity is then the small intestine. So that's where we'll actually experience that. Amylase is the one that we are familiar with. It will break starch into maltose. Maltose is a disaccharide. And that's something that we actually have taste bud receptors, and so therefore we can actually appreciate the sweetness of that. Then, once we are in the small intestine, we'll experience more amylase, because the amount of amylase in the oral cavity is actually quite small when you consider the amount of carbohydrate that we're actually experiencing. And we'll have maltase. We will also have sucrase and lactase. So these are all enzymes. ASE is the enzymes, and these are all enzymes that actually work on carbohydrates, breaking them down into smaller particles, the disaccharide for amylase, but maltase, sucrase, and lactase will break things down into their actual monomers for us. And then we will have those monomers absorbed into the cells from the luminal side. And what is the surface modification of the cells in our small intestine that will help promote lots of absorption? Microvilli. So the microvilli are where we actually are absorbing things into the cell itself. Now proteins, as most of you got, the monomers for those are amino acids. And we start our protein enzymatic digestion in the stomach with pepsin, so stomach plus small intestine here. And pepsin was the one that we learned about for the stomach. So that initiates our protein digestion. We still have protein digestion with hydrochloric acid, but I'm specifically asking for enzymes, those things that are proteins that are actually going to break things down. So we have also trypsin and chymotrypsin. So those are enzymes that we'll also encounter for protein digestion in the small intestine. Let's take a look at nucleic acids. And so we want to break them down in their various nucleotides. Because those are the monomers or building blocks of our nucleic acids. And so we will have deoxyribonuclease and ribonuclease. And so they're the enzymes that will be acting on those macromolecules in the luminal aspect of the small intestine. Lipids. Lipase is going to be the enzyme that works on our lipids, and it more specifically works on TGs or triglycerides. Remember, it's easy to abbreviate that one, TG. And what it does is it breaks it down into a monoglyceride plus the two fatty acids. And so remember, we had that glycerol backbone. I'm just going to do a 3-carbon to be the glycerol backbone. And then attached to it are 3 fatty acids. I'm going to really shorten this. And that's why it's called a triglyceride. And so what we'll have when we add lipase to this, we end up cleaving off 2 fatty acid chains. So we end up with that monoglyceride plus those fatty acids, which is really quite a different story than what we would actually expect. The majority, well, let's just say our breakdown here occurs in the small intestine. You may read that there's some other lipases associated in other areas of the digestive tract, but the amount is so small that it is negligible. So we aren't going to worry about that. We're going to just say the breakdown is occurring in our small intestine. And we must pre-treat. Before we add our lipase, we pre-treat with bile, and more specifically the bile salts of bile. And do you remember that causes that process called emulsification? Then lipase can act on that because now they're small enough. So always remember, we need to get them physically small enough for the enzymes to actually work on. And we will create these things called micelles. So we're going to also use another new term called chylomicron. And you know, you can have your own tricks for remembering where micelles are located and chylomicrons are located. But micelles are in the mucosal side of things. And so that's kind of how my own little personal trick for remembering micelles. And micelles, if you take a look at the cartoon down here, it is showing you that it is in the lumen, makes it look like a little flower, because we actually have that lipid material on the inside. We have the monoglycerides and fatty acids. So inside a micelle, it's important for you to realize, inside the micelle are the monoglycerides and fatty acids. So that's on the inside. What the micelles do is they then bring the contents to the luminal side, to actually the microvilli or brush border, sometimes it's referred to that. So now micelles come into contact with the microvilli and monoglyceride plus the fatty acids are absorbed into the cell. They can do this because they're fatty material, and phospholipid bilayer is what cells are made out of, and so fat actually then can travel very easily through fatty material, and so that's actually what happens. The strange thing, though, is once in the cell, The TGs are triglycerides are reassembled. And so those are significantly sized molecules. And so what's even more strange, but this is the way it happens, and so I have no explanation as to why. So they're reassembled into triglycerides inside the cell, and then they are then added to them cholesterol and proteins. So we're making this really strange little combination here, triglycerides, cholesterol, and proteins, and that gives us what is now called the chylomicron. So microns are still, a micron is a unit of measure, and those are really, really small things. And so these chylomicrons are really, really small things, but molecularly, we've got a lot of stuff here. And they are too big to even leave the cell naturally. And so in order to get them out of the basal side of the cell so that it can go into the capillaries and stuff, we actually use exocytosis, which you know is an ATP-driven process. And into lamina propria. And I want to put that down here so that you remember it's the basal side of the cell. These molecules, as I said, exocytosis was required to get them out of the cell because they're too big, and we can't actually get them into the capillaries because they are so big. And so what we can do, though, is we can get them into the lymphatic capillaries, hence the word lacteals, because of the milky, fatty material that's actually going into them. And so chylo, do you remember the cisterna chyli? Chylomicrons, cisterna chyli. And so maybe that also helps you to put that one into the side that actually goes into the lymphatic capillaries. So chylomicrons then enter lymphatic capillaries, because you know lymphatic capillaries are actually designed to allow large substances into them. And we call those guys lacteals. So everything else goes into a blood capillary except for our lipid-based substances, which are the triglycerides. So don't forget, a chylomicron is the triglycerides plus cholesterol plus a protein. The very last thing for our discussion on absorption here is that as far as absorption of our stuff inside our small intestine, 80% of all the material is actually absorbed before the ileum. So only 20% of the remaining nutrients in the luminal side of the small intestine are actually absorbed in the ileum. And what's really interesting, the ileum is actually the longest part of the small intestine. So the shorter segments of the small intestine, or less lengthy segments, are the areas where the bulk of actual absorption occurs. There is one thing that is absorbed outside of the small intestine, and that is some vitamins. In here are notes, they do say most of our vitamins are absorbed in the small intestine. The exception is some B vitamins and vitamin K. And that is because this is due to our colonic bacteria producing them. And since they are producing them in the colon, then these vitamins are actually absorbed in the colon. And vitamin K, remember, I want you to remember, vitamin K is important, IMPT, in blood clotting. So the only way that you can easily recognize the difference between vitamin K and potassium is potassium always has a charge with it. So vitamin K is, I can see that it could easily be interpreted as potassium, but without the charge, it's a vitamin. K plus is potassium, okay? All right. So that's the end of that chapter. And now we're going to get into nutrition. Yes?
You said that pepsin was released in the stomach and the small intestine?
You also have some pepsins in the small intestine, but the majority of pepsin is from the stomach, and then we have trypsin and chymotrypsin in our small intestine that's going to do the enzymatic hydrolysis of our proteins.
So it's released in the stomach, but it's really activated. It's activated.
It's released in the stomach and is very active in the stomach. That's where we actually start our protein enzymatic digestion.
Okay, because I was thinking for some reason that the pH of the stomach was too high for activation for pepsin.
pH of the stomach is classically what? 2.
No, I mean low.
Yes. Yeah.
But
That's what activates it. That is actually what activates it. Pepsin is produced, our enzymes are typically produced in an inactive form if they're in an organ. And in order to keep the organ from being digested by these enzymes, that's why they're produced in an inactive form. And then when you put them into an environment where they experience a pH change, that causes activation. Pepsin is actually, yeah, I don't mention it, but pepsin is originally formed, its original name is pepsinogen. And when you add hydrochloric acid to pepsinogen, it cleaves off, like acids do, because they actually also cause dehydration reactions. It cleaves off the sinogen part, and then you have pepsin, which is the active enzyme. All right.
thank you.
You're welcome. So on to chapter 27, nutrition and metabolism. I'm only saying a very short part about nutrition. What you see here on this page is all we're going to do about nutrition, because that's for a nutrition course. We're more interested in a lot of the physiology that's actually involved here. So our definition for nutrition is the study of obtaining and the utilization of nutrients for growth and life. And we have 2 main categories of nutrients. We have macronutrients and micronutrients, and the names should tell you that we need large amounts of these and smaller amounts of the others. Macronutrients are consumed in large quantities in order for us to actually grow and sustain life, and the 3 macronutrients that we talked about in first semester remain the same: carbohydrates, lipids, and proteins. Carbohydrates, remember, are also saccharides, monosaccharides, simple sugar, disaccharides, and polysaccharides. So di is 2, poly is 2 or more. But typically, since we actually have a nice definition of disaccharides, poly is, most people think of 3 or more. But anything that's beyond 1 is a multiple. Now, we have here, obviously, in carbohydrates, we have simple sugars and complex sugars fit in this category. We will find them in starch, as well as even fiber type forms. People say eat your fiber. We use our carbohydrates for fuel. Primarily for fuel, cellular energy. Lipids, hydrophobic macromolecules. Remember, these guys don't like water. That's the easy way to actually identify them. And we have all types of lipids. We have triglycerides, we have cholesterol, we have phospholipids, so there's all kinds of different categories of lipids, but we use them for energy storage, formation of cell membranes, and also hormones. So energy storage, remember that's TG, that's our triglycerides. Cell membranes, that's the phospholipid bilayer. And hormones, remember sterone, so cholesterol is the monomer that is actually utilized to actually make our lipid-based hormones. Proteins, polymers of amino acids, and these are structural and functional proteins. And so structural, typically you think of fibrous material, and functional, globular construction for our proteins themselves. So that's macronutrients. Micronutrients, we just need to get a small amount of those in, and we have to actually get them from a wide variety of foodstuffs, so not like the main categories that we actually have here. And we have this broken down into vitamins and minerals. So vitamins, small, are considered, they are a small amount that's present in our foodstuffs. And all I want you to know is the difference of the various vitamins. We have water-soluble vitamins, which are the B's and C's, and everything else is fat-soluble. And so A, D, E, and K are our fat-soluble vitamins. So that's all I want you to memorize. One of the things that's important if you are actually trying to supplement yourself with any of the fat-soluble vitamins, they also need to be taken with a relatively high-fat meal. Taking the fat-soluble vitamins with a low-fat meal actually does not improve their absorption. We also will classify our vitamins as being either essential or non-essential. And essential means that we cannot make them, so therefore we must obtain them from our diet. And non-essential, body can produce, and they are recyclable. And we're going to see a little bit more about these non-essential ones. We're going to call these that we're going to talk about a little bit more in just a moment. Cofactors are also considered part of our non-essentials because we can recycle those guys. So non-essential we can make and we can recycle. Minerals, these are essential because we cannot get them or make them on our own, so we have to get them from dietary sources. And we've talked about them mostly in ionic form. So see how many of the essential minerals that we actually have, the macro ones that we need to have in considered larger quantities than other ones that are called trace minerals that we're not going to even get into. So what is an ion that we've talked about a lot? Sodium. Sodium. So that's one of them. What's another one? Potassium. Yes. How about calcium?
Iron.
Iron is actually not considered a one of the larger needed. It's needed in more trace amounts. Actually, it is magnesium, phosphorus, chloride, 2 plus, chloride negative, phosphorus typically a negative charge, and sulfur. So those are ones. We haven't talked about much about magnesium or sulfur, but phosphorus and various molecules related to phosphorus is very important because we need that to make our ATP. So that's actually one that's much more of a molecule that, or mineral that we need in a larger amount. So you guys did pretty good on the first 3 most important ones. And it's okay that you didn't know all the others so well. But I understand where iron comes into play. but the others are actually the ones that we need more of. So metabolism now, that was all about our nutritional needs. Now let's talk about the metabolic processes that actually we utilize to use these various nutrients that we bring in. It is defined as a sum of all biochemical reactions that occur in the body, and we have two top aspects of metabolism. We have anabolism and catabolism. Anabolism, building larger molecules from smaller, and catabolism, breaking down large molecules into smaller molecules or substances. So metabolism is a combination of all of our catabolic and anabolic reactions that occur in the body. So that's what we mean when we're talking about metabolism. We're talking about making things and breaking things down. That's what metabolism is. And in anabolism, building larger molecules from smaller. So we'll use amino acids, for example, to make our proteins. I like to use this as an example because I like to always remember the term anabolic steroids. And bodybuilders like to use anabolic steroids, right? Because they're trying to actually make a larger muscle mass and more protein. So that's one of the examples I actually like to use with that. And catabolism, breaking large molecules into smaller molecules or substances. And so I like to use glycogen. And when we do hydrolysis of glycogen, we'll get glucose, correct? And we are actually going to talk about further hydrolysis of glucose into actually some pyruvates. And so that's going to be a big emphasis in actually talking about the initiation of how we're actually going to make ATP. So when somebody has a high metabolic rate, you know, I have a high metabolism or I have a slow metabolism, what are they trying to say? Where is the imbalance occurring if you have a high metabolism? You're more catabolic than you are anabolic. If you have a low metabolism, they're actually referring to that you have more anabolic reactions than catabolic reactions. So when people talk about their metabolism, you know, being slow or fast, it's basically they're referring to they actually feel like they have an imbalance of these two processes going on in their body. And indeed, some people are actually genetically designed to actually live a long time no matter how you starve them. And so if you want to live a long time, no matter how well you starve, do you want to be more baseline catabolic or more baseline anabolic? Anabolic. And so those people that say, you know, I have a slow metabolism and I have a weight problem and stuff like that, you know, you can say, well, you were designed to survive. You know, you gotta say something nice because they're telling you they feel bad, don't they? You know, so you were designed to survive a long time. So good for you. And I'm going to have to go out there and hunt and gather a lot in order for me to survive a long time, for example. Now we go into some other biochemical reactions that we're going to be spending a lot of time with the remainder of this chapter, and that's oxidation reactions and reduction reactions. Oxidation is easy to remember because you can use the word gaining oxygen. Oxidation reactions, they get oxidized, they gain oxygen. But that is not what we're going to be using with our oxidation reactions. We are going to be using the other half of this definition, which is or losing hydrogen. And not only losing hydrogen, but we're actually going to talk about plus losing electrons. So oxidation, gaining oxygen is a very basic way to remember it, but then I want you to remember losing hydrogen and losing electrons with that. Reduction is the opposite of oxidation. This is the losing of oxygen and therefore the gaining of hydrogen plus gaining of electrons. So whenever you think about the hydrogen, think of a proton and an electron, correct? Isn't that what a hydrogen atom is? It's a proton and an electron. And so this is an important aspect of that. We also will, because they occur together, we refer to these reactions as redox reactions, reduction oxidation. It just sounds better to say redox instead of Audrey or something like that, or Otter. We are going to now refer back to the cofactors, which are vitamin-based, that are the very important biochemical structures that are designed to reversibly hold electrons and hydrogen ions. And so we are going to be working a lot with NAD and FAD. NAD will become NADH. FAD will become FADH2. And so when we talk about that, right now, I want to ask you, because we have NAD versus NADH, which one is oxidized here, NAD or NADH? NAD, because when you have hydrogen, you are reduced. So right now, we're looking at the oxidized form of FAD and NAD. NAD is based upon the vitamin niacin, which is a B vitamin. FAD is based upon the other B vitamin, riboflavin. So both of these guys are going to be involved in our redox reactions that we're going to go through. And we're all interested in ATP synthesis because we need ATP for cellular fuel. And there are two ways our cells or our body actually makes ATP. One is called substrate level phosphorylation. The other is called oxidative phosphorylation. I'm going to go with oxidative phosphorylation first, and then we'll go into substrate level phosphorylation. Oxidative phosphorylation sounds like you need oxygen. Let's go. That's okay. There's nothing wrong. It's not test time. All incorrect answers are null and void in here. It's just not null and void on an exam. So oxidative phosphorylation is implying we need oxygen. And this oxygen is going to be involved in our transfer of electrons and hydrogen ions or protons. And this occurs in the mitochondria. And this is where we get the bulk of our ATP. Substrate level phosphorylation is not inside, it's not considered as part of a mitochondrial process, though we do have some actually occurring inside the mitochondria. We'll get into that detail in just a little bit. But what happens is phosphorylation, we are going to take a phosphorus group, and it's called phosphate, and transfer it from one molecule to another. And this another is specifically ADP. So we're going to have phosphorus attached to a molecule, and it's going to go to ADP and give ADP that phosphorus group, and then we'll have ATP. So that's substrate level phosphorylation. It's a transfer of that phosphate group from one molecule to another. Whereas in oxidative phosphorylation, that's inside the mitochondria. We're going to have to use oxygen to help complete this process. And then we use this other thing called chemiosmotic process. Chemi is chemical. Osmosis, remember, is the movement of water. Well, this isn't going to be the movement of water. This is actually going to be the movement of protons. So chemiosmotic process is proton driven. And so we're going to take a look now at oxidative phosphorylation inside our cells. And there's multiple steps that we are going to actually employ to get to it to make the bulk of our ATP. And these are the steps that we are going to memorize. We're going to memorize the steps called glycolysis, and we're going to memorize it as that name. And I want you to realize it occurs inside the cytoplasm of the cell, not the mitochondria. Glycolysis is inside the cytoplasm of the cell. Then we are going to have this interesting intermediate stage that will actually help us make some stuff that will give us more ATP. And that's what NADH and FADH2 are all for. NADH and FADH are the tools that we are going to use to make a bunch of ATP. So whenever you see any sort of process where NADH or FADH2 is created, realize those are the molecules that are actually going to be utilized to help us make a lot of ATP. So we're going to make sure we memorize the stages of the intermediate cycle. Then we have to go inside the mitochondria into the mitochondrial matrix for the citric acid cycle, also called the Krebs cycle, and we're going to memorize the steps of that. And then we're going to go into the inner mitochondrial membrane, or the cristae, because the mitochondria is a double membrane structure. So we're going to go into the inner mitochondrial membrane and take a look at these enzymes. All these guys embedded are proteins. They're these proteins are proton pumps, as well as an enzyme that will help generate power to pump the protons against their concentration gradient. Isn't that why we always use a pump? We actually use a pump to move something against their concentration gradient. And so what these guys are going to be is they're going to be used as proton pumps to move them against their concentration gradient. And typically, what do we fuel our pumps with? ATP. But this doesn't make sense. If we want to make ATP, we don't want to really use ATP as our energy source for the fuel for the pumps. And this is where the electrons come into play. Electrons have power with them. They are a form of energy. And so instead of ATP, these pumps use electrons to fuel them. And that's the very basic story of this. And so we're going to talk about the electrons and realize that NADH and FADH are carrying hydrogen ions and electrons. And we are going to pump hydrogen ions or protons against their concentration gradient, and we're also going to use the electrons that these guys are carrying as the fuel to power these pumps so that we can actually do this. And that's the very basic synopsis or a story about this whole process. Once we create a concentration gradient in this intermembrane space in here, you see there's a bunch of hydrogen ions or protons in here. We then use that chemiosmotic process, which is basically not a pump. It's sort of like a wind turbine or a water wheel, in my mind. It's whenever I try and think about it, when they try to describe it, that is now using the proton concentration gradient to now make ATP, because these protons will go through this ATP synthase. And when they go through ATP synthase, they actually, like water with a water wheel, they give it power to then generate our ATP. And that's the basic story there. So our mitochondria indeed are our major source of ATP production. And it is ATP synthase that is going to be the enzyme in charge of that chemiosmotic process. Let's go over the beginning of carbohydrate metabolism. C6H12O6. You should memorize that as glucose. O2, you know that is oxygen gas. 6 molecules of oxygen gas are needed for 1 glucose molecule to make 6 molecules of water, 6 molecules of carbon dioxide, and we're going to figure out how much ATP ultimately we get through all of those aforementioned processes: glycolysis, intermediate stage citric acid cycle, and the electron transport system. So let's take a look at the main phases of glucose oxidation. With glycolysis, we have a series of 10 steps that I'm not going to ask you to memorize, resulting in the splitting of 1 glucose molecule into 2 pyruvates. So glucose, which is a 6-carbon molecule, will be split into 2 pyruvates, which are 3-carbon molecules. Remember, this occurs in the cytoplasm. We do not use oxygen, and so therefore is referred to as an anaerobic reaction, anaerobic reaction. We will produce some ATP here. We'll actually produce 4 ATP, and this is substrate level phosphorylation. Because, one, we're not inside the mitochondria, so that's easy enough to remember that one. Okay. In this process, though, we will also produce 2 NADHs, and NADHs have to go inside the mitochondria for that electron transport process. So NADHs are carrying protons and electrons, and that is what we use to actually fuel those proton pumps and actually feed into them. The way that we will get, because we are producing this NADH in the cytoplasm, we have to get it into the mitochondria. And there's methods or doors or portals that the cell has in their mitochondria to allow these molecules into the mitochondria to be used. One portal is the malate shuttle, and the other portal is the glycerol phosphate shuttle, and we often call it the glycerol P shuttle. So glycerol phosphate or glycerol P. A cell, the mitochondria inside the cell will either have one or the other shuttle. So our cells don't have both shuttles. It's either one or the other way to get our NADHs into the mitochondria. The interesting thing is one of these shuttles, and we're going to go into it in a little bit, actually makes more ATP from the NAD than the other. So this is telling you not all mitochondria produce exactly the same amount of ATP, because some mitochondria have the malate shuttle, whereas other mitochondria of another cell type would have the glycerol phosphate shuttle. Why this is, I don't know, but you can actually think about this. If you're the mitochondria inside of a heart muscle cell versus the mitochondria inside of a skin cell, who needs more ATP? The heart muscle cell. So that may be a bit of an explanation as to why that actually occurs. We'll stop there and get into the intermediate step next time.